in that, chromatography technique and spectroscopy technique is one of the topmost important for the analysis of such complicated matrix or mixtures. Okay, so this is the overview of the topic. So let us start one by one. Analyte. What is meant by analyte? The component of system to be analyzed. Anything. So, for example, I want to analyze the soil sample or food sample for its pesticide residue content. So, what will the pesticides or what will the micronutrient in the soil that are called as analyte? Okay, so any material that should be determined for its quality as well as quantity are called as analyte. What is analysis? Analysis is the simple process to breaking the complex or substance into smaller part and to gain the better understanding. For example, for separation of organic mixture, I think most of the student at dissertation level, you are doing the TLC. Okay, so what will the reaction mass first is spotted on the TLC plate, and afterward we get the different separated pigments. So that is part of the pigment or that components called as analysis. And method. So method utilized for the separation or for doing the things to get separated for the analysis are called as the analytical methods. So that are the systematic methods. That should be systematic methods. It means that systematic procedure, technique, or way of doing something, especially in accordance with the definite plan. For example, we are developing a TLC, but TLC is a qualitative technique. But in somewhere in companies or in some in industry, quantitative TLC are also there. But for the analysis of the finished product like active pharmaceutical ingredients and other product, we have to develop such protocols. So each and every analysis and for each and every analysis and each and every batch, your result should be repeatedly the same, and that should not be different. And such methodology are called as systematic procedures or the technique. So we have to use the instrument. We can also use the titer metric analysis. So before going to that, these methodologies are classified into the analytical chemistry or analytical method are classified into two main categories. First is the classical analytical methodology. Second is the instrumental analytical methodologies. So classical analytical methodology having so many limitations because of the lowest limit of detection. There is no lowest limit of detection. But when you go through the instrumental technique like fluorescence, HPLC, and GC, and for ion analysis, atomic absorption, and all these things, so we can go up to the PPB level nowadays, and it is need of the current industries. Okay, so what is test method? So test method are different from the different organization like ASTM and Analytical American Society for Testing of Material. So definite procedure for identification, measurement, and evaluation of material, product, system, or service that produce a result. Okay, for example, I again take example of the analysis of pesticide residue on grapes. Okay, so as you know that residue, why it is called the residue? Because it's it is present in very least amount, and it is PPB or ppm level, and for that methodology we cannot go for the UV visible and like that. So we have to develop the technology that will capable to detect, not only detect but as well as determine the quantity that present. Okay, so here that results are very important for different organization from the materials. Okay, these appropriate methods include the before going to the analysis or development of any methodology for the analysis, we should have some protocol. So that protocol is that important thing is that sampling. We want to analyze the soil sample from specific area of Satara district, so or from specific land of any farmer. So we have to collect the samples randomly from all area which cover the all land. Okay, so that is sampling is the important step in the analytical chemistry. If your sampling is wrong, we get the results wrong. Okay, so sampling should be very important. That is important steps. And handling of the sample is very important after the sampling, especially analysis of the some food material. Sampling handling is very tedious procedure. We have to keep in specific position at specific temperature and like that. Okay, then transport. During transportation of the sample should not be changed up to the analysis. Then storage of that sample and sample preparation. So these are the important steps, are very important. so important step in the analysis of the material. So the laboratory shall have instruction on the use and operation of all relevant equipment. Okay. So if in your laboratory there are different instruments are there, so there should have instructions how to operate it. This is what is the SOPs, standard operating procedure. Okay, this is about the test method. Then minimum method information. So for suppose I want to analysis or I'm interested to analysis of the calcium content in water, so we can go for the simple titration. But I have to analyze or I want to analysis or find out the contents of the copper, lead, and heavy element in the water. We have to go for higher technique, means those technique having maximum limit of detection, sorry, minimum limit of detection. So that limit of detection can be obtained by ICP-MS or atomic absorption or industry coupled atomic flame emission spectroscopy. Okay, so method information is very important. So like that, nature of sample, what you want to analysis from the sample, that is very important to know before going to the select method for the analysis. Okay, so these are different points. I will just overlook. Appropriate identification of the method, scope of that method. For example, I want to analyze the iron content in blood. We cannot go for the gravimetric analysis because in laboratory, after taking the blood from the human body, the iron content in is very trace, and that should be determined by basic method. So that is scope, means what actually we want to find. Otherwise, we cannot find any things or any residue of the iron oxide by gravimetry. Description of the type of atom to be tested, so you should or calibrated. So what will the material to be tested? We should know about the type, parameter or quantities and ranges to be determined. Okay, so what is the range? I want to go for the ppm level, pp, that should be determined. Apparatus and equipment including technical performance requirement. I have ev, but ev not going to below ppm, so we cannot use the uv, so we have to go for the specific drug analysis. Reference standard and reference materials required. Suppose I want to analysis some contamination in food material, so I have standard. Otherwise, we cannot compare the things. Otherwise, you have to go for the IREV, NMR, and other techniques. Environmental conditions required and any stabilization period are needed. This is very important. So, all over the world, there are different regions are considered. Tropical region, humidity region. So, considering this region, environmental conditions are very important. Description of the procedure that we have discussed. The description of the procedure means affixing identification marks, handling, transporting, storing, preparation of atoms. Check to be made before the work is started. Before work is to be started, check that the equipment is working properly. So we have to calibrate the instrument if your instrument is not okay. So again, we require the standard for the calibration. For example, HPLC are calibrated by using capping. HPLC are calibrated by using capping. So it is very good standard for the calibration of the HPLC, and GC by the benzene. GC by the benzene. The method of recording observation and result. So how we can getting the data? Suppose I have taken the reading on colorimeter, and I am able to see the shift in the spectra. It is not possible. So I have to go for the visual spectrum that we can create this data smoothly by 1 nanometer curve. So we can get observation either there is shift in peak or not. Then criteria and requirement of approval, data to be recorded, and method of analysis and presentation. Once this process is allowed, so we have to record the data and method of analysis or certificate analysis and presentation. Okay, and uncertainty of the procedure. So what are the limitations of the procedure that can be included? So this is the minimum method information. So what are the standard method? Those methods are known to us or developed by any organization for different purposes. So one of the best known organization is the Environmental Protection Agency, American Society for Testing of Metal, and ISO. Okay, so appropriate method that have been published either by international organization or regional or national standard or by reputable technological organization in different publications or in different journal or in its own journals or in its own manual. Okay, so for example, you are doing practical, so your teacher are providing manuals to you how to go for the procedure. Okay, so that is how we can go for the standard method. So these are the methods already developed. We have to just carry out the experiment. So doing experiment is different thing than the development of methods. Okay, so standard methods are available. So before going to analyze develop any method, we should go for the finding the methods. So later on we will discuss. So this is the about standard method. What are the non-standard method? A new method or method not covered by standard method. Okay. So suppose anybody developed a method at Satara and I want to analyze the sample here by this method and we are getting the wrong result for the same sample. We cannot use this method and this method is not validated method. Okay. So such methods are called as non-standard method and that cannot be used even under optimal condition like temperature, pressure, what it may be the conditions of that method. Such methods are not standard methods and called as non-standard method. Methods should be developed and validated appropriately before use. That is very, very important for the analytical application. Because in food industry, suppose any industry partly doing some biscuits and they are analyzing that biscuit for the unleashed protein content, fat content, all these things. And somebody from the food industry or from APCCI, food safety, APCCI is the organization of India to control the food safety. So APCCI having its own methodologies. So you can visit the APCCI website and you can find out each and every type of the method for analysis of any type of food, means which contain the alcoholic beverages, non-beverages, etc. So all these methods are validated by that organization. So what is validation? Validation is the confirmation by examination and provision of objective evidence that the particular requirements for a specific intended use are fulfilled. What is my validation? Simple hai. What is meant by validation? Validation means it is simply the after development of methodology. We have to validate. We have to validate by varying the parameter like pH, temperature, and we have to carry out the some uh, guidelines by following guidelines like ICH guideline Q2R1. So when you join the industry, we have to go for we have to go through all these things like ICH guideline and Indian pharmacopoeia, US pharmacopoeia, all these things. Okay, so after development of methodology are validated by checking accuracy, precision, and some statistical analytical data. Okay, accuracy, precision, repeatability, LOD, LOK, and all these things. So after that, we can say that this method is valid. Laboratory shall validate non-standard method. So what are the standard laboratories? For example, ISO, APCCI. So this laboratory will validate non-standard method. Laboratory design and developed method. Suppose anyone developed a method at Satara and want to validate it, other parties can validate it. Standard method is outside their intended scope. Modification of standard method. Suppose this method is not suitable at our region or for our sample, we have to modify the method accordingly. Okay, so these are about the method. So what is the steps in method development? What is meant by method development? Method development is steps process that determine the test method capabilities through selection and optimization of analytical test parameter. So that are the parameter that I have discussed, accuracy, precision, all these things. Okay, so it provides high degree of assurance that the test method will meet or surpass the established requirement. The method development shall have been validated appropriately before use. As method development proceed, regular reviews should be carried out. So once method is developed, so we have to carry out the regular reviews. What that indicate? That indicate, suppose I have developed a method for the 10 component mixture. I have carried out the reaction in my laboratory, and there are 10 components, including our target material or our product. Some impurities are there. So during the reaction in pharmaceutical industry, suppose anybody change the starting material, or anybody purchase the starting material from other parties. So starting material is one of the main issue in pharmaceutical industry for the impurities. And nowadays in pharmaceutical industry, purity is not matter, but impurity is a major concern. Okay, so suppose anybody have changed a starting material and we are getting the 11 impurities or 11 components or 11 peaks in this HPLC. So we have to again develop method for the such. So that is called as regular review should be carried out. Okay, so here these are the three important wheels. We can method development, method validation, and method transfer. Suppose anybody have developed a method in research laboratory, and then for production of that drug on gram level. So method transfer is again critical issue for the planned scale or pilot scale application. For example, in industry in Mumbai and somebody has a plant in Goa and some other locations. So we have to again transfer the method and establish the method at that QC laboratory, quality control laboratory. So that is important wheels in the analytical method development, validation, and transfer. So these are three important steps for each and every analytical development. We are focusing on the chromatography, but we can go for invisible, pHmetry, potentiometry, each and every analytical technique. We can go for the development of methodology for the new methodology for the analysis of the any analyte. Importance of method development. Improvement in performance testing can be made by employing method development principles to standard test method when applied to new products or material. For example, I have standard method and we are actually having the 11 impurity, but method is not only showing the 8 peaks in the chromatogram. So I have to develop the method. So that is important. So we get the more again. Suppose I have method for the pesticide analysis. NRC Pune, Grape Research Institute Pune have always developing the different methodologies for the analysis of pesticide residue, because each and every year some new pesticide residues are available in the fruit sample. So we have to go for the again different methodology protocol. And once sample is tested in India, and suppose any farmer want to export this thing, and something is happened at other nation, so they people send back the sample. Okay, so why they send back the sample? According to limits given for pesticide residue, and they again reanalyze the sample, that grapes what it may be, pomegranate what it may be, any food product. So if they not satisfied or they found any some other peaks or other impurity, they will not accept your food material or what it may be your material exported by organization or any farmer or any party. Okay, so during method development process, the optimum test parameter are identified to ensure applicability and reliability of the data. Optimum test parameter. For example, I want to develop methodology for the analysis of calcium content, but calcium content in water is in variable range, and so I have to consider maximum calcium content as well as the minimum calcium content each and every bore well water, and all these things. Okay, so these are the considered for the reliability means repeatability of the data. So how can go selection of method? Laboratory developed methods are certified according to ISO 17025. So laboratory developed methods or methods adopted by the laboratory may also be used if they are appropriate for the user and if they are evaluated. The selection method. So if somebody has developed a method, we can use directly. Laboratory developed methods. The introduction of test and calibration method developed by the laboratory for its own use shall be planned activity. Okay. So many laboratories, suppose so many again pharmacy is not able to do the NMR, so they will send the sample to other organization or industry. So they have developed a NMR methodology for analysis, or they have developed HPLC methodology for analysis. So it's own plan activity, and that is the laboratory developed method. And this laboratory is certified by some standard authorization or organization called as ISO. What are the development plan? So these are the 6 plan for development of each and every methodologies. So first is the studying of sample. So sampling, as you have studied in BSc or MSc level, in analytical chemistry, sampling is important thing. Studying of the sample. So either sample is liquid, sample is solid, or sample is gas. If gaseous sample is there, very difficult to sample that material. So liquid is there, it is not too much difficult. So study of the sample, nature of sample, its pH value, pK value, nature, moisture absorption capacity, so many things are there. Each step is the method goal. So which method we have to follow, either HPLC, JC, or some other technique like potentiometry, etc. Literature review. If suppose I want to analyze of the paracetamol, if any method, suitable method is available for that paracetamol, if yes, we can go for that method. Literature review, or we can perform method by considering the parameters. So you require less efforts to develop your new methodology. Then select an approach. There are different approach for the methodologies. Optimize the method. After all optimizing para and finalize the method after validation. So these are the different parameter for studying of sample. Sample composition, sample handling storage that we have discussed. Physical properties, melting point is very important for GC analysis. Chemical properties, pH, pK value is very important for the separation by HPLC. Buffering, its solubility is very reaction with environment, responding with the used equipment and other properties. Okay, so these are the nature of sample. Then method goals. Why we are developing method? Goals define the requirements for the methods. So there are different goals. So why we are developing method? What is the reason? So there are 6 reasons. So qualitative analysis. Okay, detect the only content. TLC is the only qualitative observation, not quantitative. Then second is the quantitative. HPLC EV is the quantitative. Again, EV we are not going quantitative. We are just seeing the at which value we getting the maximum observance. Purity of the sample. For example, so many impurities are there. I want to see the purity. Characterization, for example, nanomaterial. What is the shape of that nanomaterial? What is the size of that nanomaterial? This is called as characteristic. Then purify, for example, in HPLC, preparative HPLC. We are doing column at laboratory level, but when we are going industry, there are preparative HPLCs are there, so we can directly get the fractions of the column. We need not to go develop the column and all this thing. So column development is very crucial, and we did a job, and it also get the wasted too much time and other goals also. That depends on the type of your sample and requirement of the party. Literature review. So that is one of the best methodology. You are very lucky in this area from last 5 years. You are everywhere in your hand nowadays in mobile. You can search anything on in mobile. So you can get different methodologies by literature review, like internet, some standard FDA, EPA, US FDA, Indian Pharmacopoeia, and some other colleagues and professional technique and journals. Okay, so these are different sources. From that we can get the literature or about the methods for that particular sample. Then select an approach. Okay, so what is the approach? How choose a standard test method that is appropriate for only some similar method. Okay, it is common practice to begin method development with standard test method and make slight modification improvement as needed. If standard test method is not available, it may be necessary to develop the new test method. So what is that approach? Approach means that again come with what actually I want to do, I want to analyze, I want to get. So that is called as approach. So approach is based on the different thing. Either you are using the instrumental method or non-instrumental method or classical analytical method. According to your parameter will be. Then next important is the optimization method. That is the after suppose I am developing the method by TLC. I am taking example that you are familiar with the TLC and all these things. So TLC is most common. So what I have to optimize the parameter. So what are the important parameter TLC? So nowadays TLC plate are available on the aluminum foil. So there is no issue of the thickness of layer. But if you are doing or we are preparing the TLC plate in your laboratory, we have to care about the thickness of that silica layer, as well as the slope when we putting or keeping the TLC in chamber, as well as some other humidity parameter, solvent purity. All these parameters also affects on your RF value. So RF value is the identification parameter for the identification from the RF value. You can compare the method. So this is a different method. So according to method, there are different types of parameters. So parameters are there. So that parameters are optimized during development of method. Okay. So by considering this introduction or general introduction for the development of analytical methodology for each and every type of analytical sample or any type of sample or any type of instrument or any type of the methodologies, means techniques, either classical analytical or instrumental analytical. So the most commonly used techniques in the industry are the chromatography. Out of that, most commonly used is the gas chromatography and liquid chromatography, called as the HPLC or UPLC, and most commonly called as the reverse phase, because the columns are not silica, the modified silica are there, reverse phase columns are there, non-polar columns are there. Okay. So here there are different types of the technique. I will not go in detail the techniques. You can read from the. So we'll focus on the HPLC. So this is the typical instrument of the HPLC. So these are different components. So first component of that HPLC. HPLC is very sophisticated analytical tool nowadays. 90% used for the pharmaceutical analysis and all other organization, because it's separate as well as quantified. It's not just a separate. So here we can separate different types of the material, and there are limitation of that technique, both HPLC and GC, where the some samples are not suitable for HPLC, we have to go for GC and vice versa. Okay. So solvent track or reservoir, pumps, injector port, separation column, and detector. So heart of the that method or HPLC chromatography is this column. So column chemistry is very important. So this, by considering all these components of that instrument, we have to optimize the parameter during the development of method. So these are the one developed method. So column. So there are different companies, Thermo and Steel, and so many companies. When you join the company, we can have idea about different types of company and how to read the column. Okay. So this is column and this is the method. Suppose anybody in your industry, you hand over the sample, and he, at the same time he will give the method. So that is the method, and you have to do analysis, or you have to go for analysis of the by considering or by using this method. So at that time we found such type of parameter conditions. So which type of column should be used, which type of mobile should be used, what is gradient isochromatic, that might be I think you have studied in your MSc course. Flow rate of the mobile phase, pressure of that pump, because these are the fully automated machines or instrument. Then temperature and injection volume. It's very small amount of samples required, and detector. So there are different types of detector in HPLC. So this is the method after validation, after development validation. So such methodologies have to be developed for your sample or any drug in your laboratory. Okay. So there are several reasons so that we have discussed. This for specific for the HPLC or for chromatography. Suitable method for particle analyte in specific matrix is not available. So why need to go for method? For example, we have discussed I want to see the number of components, but existing method is not capable to separate and identify. We have to go for the analysis or develop the method. Existing method may be unreliable, having poor precision or accuracy. That is very important terminology: accuracy and precision of the method. Okay. Repeatability of the method also a major concern. So these are simply we can say the accuracy and precision. Existing method may be too expensive or time. For example, we college or our university or our institute, we have not any machines or instrument, and I want to go for analysis of water, and we have to develop method on AS. So we have no ICP-MS, so we have to go for the development of methods But given method is ICP-MS, but simultaneously we can go for the other methods if possible. And if traces of heavy metal, for example, mercury is there, copper is there, and lead is there, such type of element cannot be analyzed. But if it is in available in different water sample at trace level, it not be detected or it not be observed in your graph or in spectra by using AS. That is, I have just example. So here, overview of the method. So for HPLC development, define method goals and type of chromatography. Okay, so which type of chromatography? There are different types of chromatography starting from TLC paper up to the LCMS, GCMS, and some high penetrate technique like that, and some other advanced technique are very special for the biological sample like exclusion chromatography, gel filtration, etc. Size exclusion. So the established sample preparation process, that is very, very important part in development of methodology, because collection of sample and after treatment of sample. For example, I want to analyze the pesticide residue in grape. So I have to critically treatment the sample. We have to go for the extraction, crushing into fine powder, then extraction, then remove all other impurities, and then in separation by separatory funnel, and finally we have to inject in a HPLC or GC column. Select mode of separation, either column, mobile page. Okay. Uh, Perform preliminary separations. So sample is very complicated. We have to perform the separation. If sometime samples are not ready to detect, so we have to go for derivation, prepare the derivative, optimize the conditions. So all these conditions like temperature, pH, column type, column stationary phase, mobile phase, viscosity and polarity is very important, and validated. So these are the overview of the method development strategy. So these are the goals I have just overlooked. How many analytes of interest? That is very, very important. Actually, when sample is unknown to you, we have to go for trial and error basis. Okay. So how many analytes are present? Somebody told you uh, in this mixture or in this sample 10 compounds are there or analytes are there. So we can, after injecting the sample, suppose we get the 10 peaks for 10 sample, there is no issue. But if sample, we have no idea about how many analytes are there, so we have to go for the, we have to critically think about that. This helps to make choice of efficiency and resolution of the method. So for food pesticide residue analysis, in single run, we can separate the 70 component, 50 component, 70550 in a single GC method. Okay, so at that time we cannot go for any other method. Only GC is the technique. What will the sample matrix? Is there is need to separate analyte from many matrix now. Matrix, what is meant by matrix? I have taken example of grapes. So apart from the pesticide, there are so many things in the grapes or any food material or any sample. So that's called as matrix. For example, biological sample, urine is there, blood is there. So there are so other components are called as matrix. Require special sample preparation steps such as a solid phase extraction. Solid phase extraction. Is the method qualitative or quantitative? Is this method is qualitative? Suppose TLC is qualitative, our interest is not to go for quantitative. We do not, we cannot think about the quantity. We have to only focus on the qualitative. Either present, either there is compounds are present or not. We can tell. So these are the goals. What is the concentration of analyte? We may need to deal with range of analyte concentration. Is there is a need of high sensitivity separation detection. So we have discussed about. Suppose I want to go for lowest detection limit. So for example, if there is adulteration in food by some companies or some manufacturers, so I have to, or we have to find out adulteration. We have to go for trace level, because adulteration is always at a trace level. Mixing of some other things in a food material. Okay. Analyte sample matrix, solubility and stability. Suppose I am using the water as solvent for some drug, and after 2 hours, drug is not stable in that water. So at the same time, considering solubility, we have to also check the stability. We need to exclude certain HPLC technique when dealing with unstable or sparingly soluble compound. Okay, so important criteria for the separation is compound should be soluble in solvent or water or buffer. Okay, so these are the different parameter separation goal in HPLC method development. Resolution. Okay, resolution means separation of two peaks that should be greater than, these are the parameter you can read from the, there is a very good book, instrumental method of analysis by Scoop. So we can find out some all these basic criteria in the chromatography. That should be greater than 2 or it will be 1.5. Separation time, I am doing some method, I am developing some method, it takes the 2 hours or like that, so that is not suitable for, or that is not economical or suitable for industry. Okay, so that should be up to 30 minutes, it's okay. Quantification, assay, quantification, if method is developed method is sometime we getting 98% purity, sometime we are getting the 92% purity, so quantification is very important. So that RSD value, relative standard deviation, but precision of the method is very important. Then peak height, if our UV sample not showing any peak, so we cannot get the lambda max values. More similarly, HPLC, peak shape is very important. Gaussian peak is very important for calculation of the concentration, because in chromatography, peak area under curve is a direct determination of the quantification, determination for the analyte presence in the sample. Solvent consumption. So acetonitrile, methanol is one of the most commonly, they are the most commonly used solvent for the pharmaceutical industry or for HPLC as a mobile phase. So that should not be minimum mobile phase use per run is means we have to minimize the time. So what is known about the sample that we have discussed in last some slide? So like chemical structure, then acid groups are there, basic groups are there, molecular weight. If high molecular weight is there, we have to go for the if we are going for analysis, and GC we cannot analyze the sample having high molecular weight, so we have to go for the HPLC. Stability, either it is photostable, solvent stable, that we have discussed again. Solubility of the sample in different solvent or suitable mobile phase. Concentration of that sample in that in that mixture, and matrix, other interfering substances. Okay, so sample preparation is one of the important step in the HPLC method development to get the reproducible result. Okay, so after taking some milligram of the sample, suppose I want to prepare 100 ppm of sample, so at least 100 ppm sample should be soluble easily within 10 minutes. Otherwise, we have to go for sonation and heating. It is not suitable. After heating, again it will precipitate out. Okay, so this is very important. For sample preparation, sometime if complicated materials are there, we have to go for the extraction, all these things. For example, analysis of the element by AS, we have to firstly treat the sample by using the different types of acid, and then we can send the sample. So more similarly, HPLC, if drug samples are there, there is no issue. But in some food samples are there, some agriculture samples are there, soil samples are there, we have to treat the sample to remove the interference or to remove the some particulates available in the sample. These are the samples preparation step. We have our balance should be very accurate, because weighing balance is very important, having a precise and repeatable result. So weighing and volumetric dilutions are important key parameter during sample preparation. Sample extraction if required, filtration in some because HPLC columns having very small diameter of the that column, so we have to filter the sample, centrifuge the sample, and solidify. So why we need to do this thing? To remove the interference, and derivation. So derivation, one of the technique used if we are not getting any type of response of that peak in a chromatogram, so we have to go for the derivation of that sample. So derivatized product, we get the response in the HPLC. So suitability of the detectors. So which one is the commonly used detector in HPLC? E-visible, or we can say the photodiode array. Nowadays is called as the photodiode array. So such type of detectors are used. So along with that, some hyphenity technique. Nowadays HPLC, GCMS are there. So most commonly used detectors are E-visible. So then repetitive index is universal detector that depends on the polarity and thickness of that material. Evaporated light scattering detector used for the protein sample analysis. Fluorescent detector is very good, but sample should be fluorescent after excitation of some light. And electrochemical detector sample should be having capacity to oxidize or reduce. Okay, so this is the selection of detector during the development of methodology for the sample. So you have to go for the different detector and then you can find out the so detector selection is depend on the chemical nature of analytes and potential interferences. Suppose someone or some compounds is observing at 270 nanometer, at the same time some other impurities are going to observe, so there is no use. There is a no use. So because of that reason, such overlapping materials are not allowed. So at that time we get the same compound. So for that reason, we have to potential interference are also considered a limit of detection required. But what up to depth or up to limit we want to either ppm, ppb, ppt. Ppt is only for heavy element and some like arsenic, all these things. Availability and cost of detector. Suppose we want to purchase the machines and detector cost is detector is MEMS, so it is not possible. Instead of that, we can purchase the e-visible supported HPLC system. Okay, so here this is the diagram for the EV detector. When sample come out from the column, so here light is allowed to fall on the sample. So once a sample that is eluted from the column move along or come along with towards the detector. So here at this point, this sample will get a uh, light will get observed by the sample and we get the peak in the chromatogram. Okay, so suppose any sample having a literature data is about 300 nanometer, but in different solvent at different pH, there is some slight changes in the shifting of either blue shift or red shift will be there for the that lambda max value. So that should be considered. If these are shifting or these things are shifting 10 nanometer like that, it is not acceptable. Okay, so another one thing is important thing is that you during UV visible detector, most of the solvent absorb the UV light. It is called as UV cutoff. It is called as the UV cutoff. So UV cutoff of the ethanol is 205 nanometer. UV cutoff of the acid nitrile is 190 nanometer. So what is UV cutoff? So below that value, for example, 205 for the methanol, ethanol, and 190 nanometer for acetonitrile. And because of that reason, they are using most commonly for the analytical development. So what is that reason? So most of the our samples organic compound absorb at 250 nanometer, 254, 70 like that. Okay. So solvent will not absorb in this region. So where solvent will absorb? Below 190 nanometer. So that is what you use. So solvent should be important during the selection of the detector. Alternative detectors are available if sample is not UV active. So when we have to go for the alternative detector, if suppose hormones are there, some compounds, organic compounds are there at that time, that compound does not contain any chromophores. At that time, we have to go for the other detectors. There are different types of detector. Mass detector that is based on the ionization. So by considering the mass detector, sample should be ionizable and at desired pH. So for basic compound, we have to adjust the neutral to basic pH. For acids, and for acidic pH 3 to 4 are used. So during HPLC preparation of buffer, we have to also consider, we have to use the neutral buffers. So what is that neutral buffer? Neutral buffers means not neutral, but the buffers are non-salty buffer. So sodium phosphate means salts of the phosphoric acid are most commonly used for the pH adjustment in HPLC method development. But in this case, we have to use for the ammonium formate formic acid, because in mass, the organic compound form the sodium atom. Suppose acid group is there, so there is formation of sodium or potassium salt, and that mass of that potassium or sodium is also considered. Because of that, during selection of mass, we have to again change our mobile phase. Okay, so there are different types of the buffer systems we have. Then repetitive indices. So this is the physical property. So here But this method is a this is actually universal detector, but all the methods are fails, we have to go for it. But we are getting sometime wrong result, and repeatability is not good, means uh, precision of that method or developed method is not good, because when small change in the mobile phase, there is change in the RI value, and because of that, such detectors are used only when there is a urgent need or there is no any options for that sample. Okay. Also not used for gradient. Gradient means, what is gradient? Gradient means there is change in the mobile phase concentration according to the time. Suppose acid nitrile water 50-50% zero time peak. After that, one by one, that going to change with the speed time. So changing the mobile phase concentration of different solvent, there is change in RI. So this is only for the isocratic elution system. Isocratic elution system. Then detection, detectation fluorescence. So simple criteria is that this is very sensitive detector, but condition is that sample should be fluorescent, or it able to emit the light after excitation. Okay, it is similar to the invisible detector. Then last is the electrochemical. This is less commonly used, and again for only sample having a oxidation reduction capability, or can be oxidized purposefully during the relation. So this is the sample from the column, and that is a uh, electrochemical cell. So where we can get the signal and the peak. Okay, so this is about the data selection. Selecting mode of separation. Okay, so might be you have studied uh, in your last 2 to 3 years in across the chromatography technique. There are two important mode of the chromatography. First is called as normal phase chromatography, and second is called as the reverse phase chromatography. So in normal phase chromatography, uh, your stationary phase is polar, and mobile phase is non-polar, and for reverse phase, stationary phase is non-polar, and mobile phase polar. So why HPLC technique are most common? Because reverse phase is non-polar. Sorry, in reverse phase, stationary is non-polar, and most of our solvent are polar, and most of our compounds are soluble in organic solvent or polar solvent. If you are using normal phase, we have less chance to dissolve the sample, because sample should be dissolved in your mobile phase, and then it gets separated. Okay. During selection of mode, nature of sample and solubility is very important, that we have discussed. Okay. Then this is the reverse phase, that is the different phases are used. So octadecylsilane. So what is reverse phase? Reverse phase means silica are modified, SiOH are modified by using the straight chain carbon alkane compounds. So called as C18. C18 means octadecylsilane. Octyl, C8. Okay. Butyl, phenyl, phenylhexyl, and PF. Okay. So most commonly used columns are the, this is the sample, this is the reverse phase system, where non-polar systems are there. So why it is non-polar? Even silica is a polar one, but it is modified by the long chain C18 alkane chain. So this is called as polysiloxane. It's called as polysiloxane, and it is called as commonly called as C18 column or octadecylsilane. So because of that hydrophobic alkane chain, so this material, this column stationary phase becomes a non-polar. So once stationary phase is non-polar, we can use the polar solvent. Okay, so during separation or distribution of sample in between two different phases, there is a one should be polar and another should be non-polar or mid-polar. Okay, so this is the about reverse phase. So this is the interaction of the material with the stationary phase is a non-polar. Okay, so interaction is that hydrophilic material. Hydrophilic means that leaving the mobile phase, because in reverse phase chromatography, mobile phase is polar. So hydrophilic will come fast from the column. After that, medium polar and hydrophobic materials will come, because your column is a non-polar. So here, in normal phase chromatography, we have to use the silica column. So I have shown here silica, uh, silica modified with ammonia group, like cyano-silica aminodiol. Okay, means all polar groups are there. All polar groups are there, are called as the amino-cyanodiol. Okay, so here in this case, stationary phase is a polar. So this part we have studied in HPLC, but here what we have to consider, we have to consider the nature of sample and what column should be selected. This is we are talking about the development of analytical methodology for the any unknown sample or unknown mixture. Okay, so in this case, mobile phase should be non-polar one. Okay, so what the mobile phase are used like isopropanol, hexane, and combination of ethyl acetate are used. Okay, so this is most common practice for the TLC. Ethyl acetate, hexane are most commonly used mobile phase for the TLC because our TLC plate coated with the silica gel. Okay, this is called a normal phase. Mode of selection. If some ion exchange concepts are there, at that time we have to go for also ion exchange type of the thing. So what is that ion exchange? When analytes are ionic, for example, uh, like our atomic absorption spectroscopy, we can also go on this for sulfate, calcium, magnesium, cations and anion by ion chromatography. It is called as the, it is more similar to HPLC, but column is capable to exchange the ion. Okay, so that again the strong anion and cationation that we have studied in ion exchange chromatography. Okay, but ion exchange is different thing. Here ion chromatography are they developed based on the conductivity detector. Okay, so such used in amino acid peptides samples where uh, materials or sample or analyte having a charge. But as you know that, sorry, as you know that for amino acids, pH is very important because amino acid contain both the charges are possible at its acidic point. Means at the acidic point, the material having a amino acid having both the charges, positive as well as negative because of the amino and acid group. Then reverse phase system ion pair formation. So the ion exchange is different. For example, this is the phenyl propenylamine and it is quaternary ammonium, quaternary nitrogen is there. And if quaternary, because your drug, uh, you have observed on different drug are named as hydrochlorides, etc. Why this is because of two such for such sample we have to add the ion pairing reagent during the development of methodology in a mobile phase. Okay, so octane sulfonic acid. So what are the ion pairing reagent? Ion pairing reagents are, suppose your analyte contain having positive charge, we have to add the negatively charged ion pairing reagent. So alkyl sulfonate. And if your negatively charged sample, we have got the quaternary ammonium based ion pairing reagent. So this is the mechanism. By this is the column core. This diagram is the column core, and we can get very good separation by using the ion pair formation. Because keep in mind, in chromatography, firstly we have to retain the sample on column. We have to retain or absorb the sample on column, and then we can successfully one by one separate from the column and then detect it. So this is the graph. There is no ion pairing reagent added. 3 peaks are there, but when we add the ion pairing reagent, we get the 5 peaks. So that is how the chromatography techniques are developed. Sorry, methods are developed. Size exclusion is used for the size exclusion is the technique based on the protein sample having different molecules. Okay, so gel filtration, size exclusion chromatography having different types of testing phases are used according to the size of that protein or biological molecule. Why is the reverse phase is most popular? Why is the reverse phase is most popular in the development? Everywhere you can see reverse phase chromatography technique is the most commonly because large proportion of analyte are water soluble that we have discussed. Um, uh, our sample should be soluble. Most of the sample are soluble in water. Wide range of stable stationary phases are available. Okay, so here I have shown this is the stationary. So we can modify this stationary phase accordingly as per the requirement. As per we can increase or decrease quality from silica, simple SiOH to hydrophobic stationary phase C8 C18. Okay, then here we are. Okay, sample mobile pages work for many applications like water acid nitrile. I have because water is used to make the buffer, and acetonitrate is to modify the polarity. Okay, then retention and solidity are altered by changing. So by using the reverse phase mode, means stationary phase is a non-polar, we can alter different thing. So what is chain length can be altered. Mobile phase, organic solvent type, different mobile phase combination, 10 90 10 50 50 70 30 etc. we can make the combination. Percentage organic, pH, buffer and other additives can vary, like ion pairing region. Temperature, especially with ionizable analyte also. Yeah, so because of that, most important criteria that we can modify the different thing according to your sample, because all the samples are not same. For example, hormones are there, some other pesticides are there. Pesticides are have, most of the pesticides have no any chromophores. Okay, so for that we have to go for the GC or some other detector in HPLC. So general rule in mobile phase selection is that in partition chromatography, uh, the selection of mobile phase depends on the like enhanced separation, so sufficient polarity, improves the peak shape and alters selectivity. So selectivity is the factor that is called as alpha, and that is related to the resolution of peak. Resolution means there is this clear-cut distinction between the two peaks. There are different types of mobile phase are there. So most commonly used and their EV cutoffs. So isopropyl alcohol having somewhat more density. If high density solvents are used, there is a back pressure in the column. So because of that, we have to use a less high polar, having a less EV cutoff value as well as the less density. So such solutions are acetonitrile, methanol, and THL as a modifier for the polarity of the mobile phase. Okay, so these are the water miscible, low viscosity, low UV cutoff, and unreactive. So this is the what happens when we change the solvent. So for the same, this is the typical chromatogram with respect to time. So there are different peaks are there. 40% methanol is there. If 40% acetonitrile is there, and 40% THF is there. So by using 40% methanol, we getting the very good result separated components of the mixture. Okay, so that is the good separation. This is also, but at the starting there is merging of peak, so there is not good resolution. Okay, again, suppose we modify the percentage of the polarity. 100% acetonitrile is there, 80% acetonitrile is there. So what happened? There is only one peak. Okay, but when we changing the acetonitrile up to 45%, we have get a good separated peak. There are 5 components are very well separated. Okay, so for saving time, we have to consider, or we have to consider both the things. The time separation time should be minimum. At the same time, peak should be resolved very well. Okay, then next part is the isochrotic and gradient. So what is isochrotic and gradient? So isochrotic is the changing the mobile phase composition. For example, acetonitrile and methanol is there, and there is change in mobile phase composition according to time. It is called as gradient. But in isochrotic, there is a constant composition of the mobile phase across the time. Okay, so in this way, we can go further. Either we have to select the isochrotic mode, or we have can select the gradient mode. But this is also depends on the type of mixture and how many compounds are there. Okay, more compounds are there, and isochrotic. We can see here this peak is eluted out or compound eluted at 6 minutes, but by using the gradient, we have the within a minimum time. Okay, so there are different gradient variables are there. Successful gradient. So this is the how gradient. Okay, so increasing the, for example, 50% is acid nitrile water, so we can decrease one by one. So this is systematically applied by the machines or by the software. Automatically pump will mix the components of the mobile phase and resolve it. Okay, then there are different disadvantages that we have discussed. Gradient require the multiple reservoir as well as pumps, and gradient uh, require more time to analyze, because at the starting there is less amount of at the starting there is a different proportion of the solvent, but at the end there is a different proportion. So we have to restabilize the column and then go for the analysis. So it is firstly longer run time is required. Okay. Second is the column re-equilibrium. Okay. So during method development we have to, if we can develop method with isocratic one, we can go for the isocratic. Okay. Then column selection is very important. So column chemistry we have discussed the reverse phase, and what is the, so more carbon, more hydrophobic phases are required. We have to go for the C18 type of column, and if mid-polar columns are required, we have to go for the C8 column. So this is again depends on knowledge of sample, pK value of sample, which are the functional group in the sample, properties of the column packing material, and how it compounds will interact with the packing material. So what is the interaction of that compound with the packing material? Okay, so that part we have discussed here. Silica based columns are most commonly used. 75% columns are silica based. So octadicyl silica, C8, phenyl, phenyl hexyl, etc. are the column. So silica is there, but this silica is modified using the different types of the modifier. Okay. So this is called as packing material is very important. So we have to select the column. So column is very important. And another one important packing material. So this is C8 column and C18 column. So might be you have studied Venn diameter equation that gives the column efficiency and packing of the column. So more packing materials good give the good plate height. So plate height and ED diffusion, longitudinal diffusion, and mass transfer. So these three things are given by the Venn diameter equation, and this is very, so this HETP, height equivalent to theoretical plate. So efficiency of column is depends on these three parameters. So column packing should be uniform grain size, and it should not it's having proper functional group and all other parameters that I will not go in detail, but we have to keep in mind. Suppose there is no uniform particles, so we get the band broadening or broad peak are there, but uniform particles has get the smooth or sharp peaks. Okay, so all these parameters are very important. We have to optimize the parameter, and this is called as the packing material. So we need not to add the packing material. Already columns are available in market. Column dimensions are really important. So more commonly used columns are having a 30 centimeter or 300 millimeter, and short column is about 30 to 50 millimeters, 3 to 5 centimeter. Okay, so there are different ranges of the column available up to 10 to 12 millimeter high sample loading. So according to column, we can also get the different types of refrigeration, but we have to keep in mind if sample loading is high, we are injecting 100 microliter of sample, and more concentrated sample, your sample should be, sorry, column should be long. Okay, so what will the compounds we added that should be retained. Suppose we are using very small column, so all the compounds will come immediately from the column, so we get the broad peak. So when using the small column, we have to use the small amount of sample, and then pK and mobile phase. So that is very important. So pK, pH equal to pK by Henderson equation. So we have to keep, for example, benzoic acid is there, we have to adjust the pH below its pK value. So that should not be dissociated. So benzoic acid completely in a undissociated form. Okay. So for example, acid is there, low pH, most retention means that will retain on the column. If ionic species is formed for that acid, at pH equal to pK, broad peak is there, and if completely ionized phase is there, little retention means compound will immediately come out from the column. Okay, this is about the mobile phase selection, about the pK value and pH of the mobile phase. Okay, so this is very well known equation to you. pH equal to pKa. So most of the buffers are used are phosphorus buffer because phosphoric acid have different pK values, and we can use a different pH up to the 12. Okay, and some citrate buffers are used. Some other buffers like trifluoroacetic acid, phosphoric acids are also used, and ammonium buffers are used for the LCMS system. Okay, so these are the different types of the buffer systems can be utilized. Then another important parameter is that temperature control. Okay, so what is that temperature control? Temperature control is the when you viscosity is changed by increasing temperature. Okay, so in this case we can see at 25 degrees Celsius we have 20-minute run time, but when increase the temperature we have the minimum retention time, but there is merging of heat. Okay, so this is happened because so we can select the 35 degree as the column temperature. So this is all about the methodology, and after this we have to calculate the parameter, and then from that HPLC basic parameter, validation parameter, we have to decide either this protocol is implemented or not. So important is that retention factor are, you can say the capacity factor, then selectivity, efficiency means number of theoretical plate, and resolution. Okay, so this is the typical spectra, HPLC, sorry, typical chromatogram for the HPLC. So in this case, these four important things are there. Resolution means good separation between two peaks will be there, and resolution will be more than the 1.5 value. So width of the peak and retention time of the peak is considered. Then column efficiency. What is column efficiency? Capacity of the column to separate or get the very good resolved separation of the chromatogram. Okay, so this is the Gaussian peak, and this capacity depends on again when temperature equation. According to packing material, length of column is a direct relation with the length of the column as well as resolution. Okay, so we can increase the length of the column for a more number of the theoretical. Then this is about the again comparison of the column efficiency based on the theoretical plate. So peak should not be broad, so n will be small. Okay, so if more theoretical plates are there, good efficiency or good resolution or good method is there. Okay, means method having good separation efficiency. Okay, then another important parameter is that retention factor or capacity factor. So this is calculated by using the wide volume. What is wide volume means at time of injection T0 and retention time of the first peak. Okay, so in this case we have to just a minute. Okay, so in this case, resolution is also important. Resolution of the components depends on the capacity factor and retention factor. Okay, so that should be very good separated peaks are there. Then selectivity of resolution. Okay. Then selectivity and resolution, and these are the some important parameter that we have considered during the selection of the parameter applied on the selectivity of the reverse phase HPLC technique. Okay, so here some other parameter like peak shape, tailing, and fronting are there. So that will not show, but peak should be will be good. Fronting means tailing means there is no sharp peak. Fronting means at the fronting there should be some non-Gaussian peaks are there. This is called as the asymmetric factor. Okay, so this is all about the method development. So these parameters are considered during the method development, and after development of method, we have to validate the method for the be according to different techniques like standard organization, the British Pharmacopoeia, US Pharmacopoeia, and international pharma. So these are like precision, accuracy. All these things are considered. Okay. So this is all about my laboratory and department, my research at UTU. This is campus. I acknowledge to my university and also acknowledge to ICI Satara for inviting me for this lecture. And Anil, thank you.
Okay, thank you, sir.
संवत्सर आहेत का संवत्सर आहेत का मॅडम
चव्हाण सर. नाही येत होते. Okay. Now may I request Dr. V.B. Ghanwat sir to give a vote of thanks.
Okay, it is my immense pleasure to give vote of thanks for this lecture. I am very happy to say I am a student of Dr. Anil Gore sir.
Thank you.
I know whenever I was in MSc, Dr. Anil Gore sir, he was doing his research work, and from that time he taking our practicals, and I was very eager to take lecture or arrange attend the lecture of sir. Today I got this opportunity. Thank you very much, sir. You have given all the points of this analytical chemistry, and I know that all these questions these are asked for analytical chemistry or whenever we go for the company interview as well as very any research institute. All these points, all complete whole this lecture it is covered or whole these questions are asked for that interview. I have given lots of interview, and during listening this lecture, I remembered all that interviews also. Thank you very much, sir. You have covered all the points of analytical chemistry related to research as well as for company purpose. Thank you very much. I would like to
Thank you.
thank our honorable principal, Dr. B.P. Jadhav sir, for giving opportunity for arrange this lecture. Then on, I would like to thank our head of the department, P.V. Bise sir, who have given us a permission to arrange this lecture for both MSc first and MSc second year students. Our MSc first year students, they are listening this lecture on another link because for Google Meet we have only 100 limitation. Then I would like to thank our coordinator, Dr. Santosh Kamble sir and Dr. P.V. Chavan sir, who have been arranged this lecture. And I would like to thank Vishaka Walekar madam, who have anchoring or whole throughout from the morning for this lecture. And at last, I would like to thank all the students who have been patiently listening this lecture. And with this, vote of thanks. With permission of arrange, I would conclude that this lecture it is over for now. Thank you very much to all of you. Thank you very much.
Thank you.
Thank you, sir.
If anybody, sorry, if anybody have to ask any student they have any question, if they have, sorry, oh, sorry. Anybody have any doubt? कोणाला काही डाऊट असेल तर विचारा प्लीज. Okay, sir. If you have any doubt, you can also text on our. I will forward it to Dr. Anil Gole, sir. Then we will discuss about this. Okay. Thank you very much, sir. Thank you.
Thank you.
Okay then, thank you.